This video demonstrates our new fully automatic approach to computing biological pathway maps in urban map style. Biological pathways, structure depicting our knowledge of molecular reactions in all types of biological systems, have been extensively investigated and lead to a complex hierarchical relationship network beyond human reading and drawing capability. The present approach systematically untangles video clutter by decomposing large networks into semantic subnetworks and bundling informative undirected edges to create space for bidirectional or unidirectional relationships. This is designed based on biologists' preference to investigate detailed relationships between chemical components while simultaneously exploring the roles of them, such as ions, antioxidants, or cofactors, to image how the changes of chemical components could affect the entire physiological system. We automatize the manual drawing process of biologists by first partitioning the map domain into multiple subblocks and then building the corresponding pathways by routing edges schematically in order to maintain the global and local context simultaneously. Let us give the major human metabolic pathways as an example. Glycolysis is the essential metabolic pathways converting glucose to energy, where the preparatory phase first consumed energy to convert the glucose into 2,3-carbon sugar phosphate, and then the pale phase produced the energy currency ATP. Using our layout, we know that the process only occurs in the glycolysis, gluconeogenesis, to provide the local context to the glycolysis pathway. Another well-known metabolic pathway is the urea cycle, where our body eliminates the toxic ammonia by producing and discharging urea in our physiological system. The entire process converts ammonia to urea by consuming the energy stored in ATP. We therefore follow the movement of ATP and explore how to bring ammonia entering the urea cycle by converting it into carbomoyl phosphate and further citrulline, so that citrulline can be brought from the mitochondria to the cytoplasm through the transport pathways. In our visualization, this process is clearly shown that these chemical components are moving from the urea cycle to the transport, to the alanine esterase metabolism, and back to the urea cycle to provide the global context to the urea synthesis pathways. The algorithm is done by introducing new constraints to the floor planning problem to have better control of the block placement and distributing dense edges using floor network algorithm. The proposed design allows us to enrich visual coding for a small pathway network and scale up the conventional orthogonal layout algorithm to lay out a large graph such as human metabolic network. In our system, we also allow users to constrain the block placement by specifying their preference to retain user experience on the layout. This example shows the dataset from Cake Overview Map, where we automatically place blocks that share more chemical components close to each other. Metabopolis successfully groups the categories with warm and cool colors together as originally designed. Users can also edit the relative positions between categories to generate a more cake-like representation. To support data exploration, content adaptive navigation together with highlighting target of interest and their corresponding neighbors are finally incorporated.